All right. Can you all hear me? Great. So you've got some data, and the data are going to have patterns, because that is what data do. They have patterns. How do you know whether these patterns are real signal or whether they're batch effects and noise? And here Feynman very wisely tells us that we have very powerful ability to see patterns. The human mind has very powerful ability, and it's easy to see the patterns that we want to see. And the gist of my talk is going to be summarizing some of the ideas that we have as to what are useful ways to distinguish false patterns from correct ones. And the other thing that is very important to say in this context is that there is more at stake than fooling yourself or me fooling myself. What is at stake here is whether we'll establish this community as a trustworthy source of quality data, or whether some early haphazard judgments can tarnish the burgeoning opportunities that we had ahead of us. So I think it is very careful for all of us as we generate and analyze data to be very careful about the inferences that we make and how well supported they are by the data. And this is particularly true if you think that your data are so good that they don't need quality control. That means that you really need to have some QC measures so that you can establish whether the data are indeed so good. And we've already shared with you some of our ideas as to how you might be able to distinguish single cells that contain analyzable signal from those that are most likely to contain background, noise, co-isolation, and so on. I should say that we have tried a dozen more metrics in addition to these that we finally ended up using, and there is certainly space for trying even more. We found that the metrics that for us worked very well were the signal-to-noise ratio that we measure for the reporter ions. And as Alex pointed out early today, it is very important to keep in mind, to put in perspective what that signal-to-noise ratio is, because if that is 10, then you cannot estimate fold changes larger than 1 to 10. Or if it's 100, then you can estimate at most 100-fold fold changes. What are the limits of quantitation given the data that we have? And we have found this coefficient of variation for relative peptide levels that most of you coming from the mass-spec community are well familiar with, to be extremely useful in being able to discern which are the successful and the not so successful cells. A highly related rubric to performing QC is to evaluate the reliability of the data, because it might be that some wells did not even have a single cell. We did not sort it, and therefore they're not usable. We are going to remove them. But also, the cells that provide useful data might have different quality data. Some of them might give us more reliable data points than other ones, and we have ways of assigning confidence intervals and reliability to individual measurements, which is incredibly useful. We cannot do this with single-cell RNA sequencing. We cannot have an error bar for how much we trust the reads for a particular gene in a single cell, while here we can assign confidence and reliabilities to individual proteins in single cells. And we may want to weigh our data differently. We may not just want to put them in the same pile of data points, but we may want to assign different weights. And here, two related concepts of evaluating first for batch effects and then trying to correct for them and integrating multiple data sets. Every high throughput method that they've heard of has batch effects. That was true for DNA microarrays. That is true for RNA sequencing. That is true for single-cell RNA sequencing. That is true for bulk proteomics. It's no different for single-cell proteomics. But rather than just try to correct for the batch effect, I also find it very useful to see how much of that there is to begin with. Because one thing that I have found over and over again in our data is that when our chromatography is performing great, batch effects are very small. when we do the experiment well, there isn't much left to correct. At the end of the day, we may still have to do the correction, and we like using Combat, which is an Eve-based framework for doing it. But I think it's also very important to ask yourself, what would the data indicate without that correction? How am I correcting the data? And what is the best way to do it? The methods that we use are methods that were developed for DNA microarray, and they're microarrays data, and they're applicable, but they don't take full advantage of unique features of proteomics data, and there's certainly opportunity to design methods that are better suited. And the same goes for integrating multiple data sets, not just multiple single-cell protein data sets, but integrating single-cell RNA sequencing with single-cell proteomics data and so on, and doing this intelligently. There have been a lot of emphasis and a lot of effort to do this for single-cell RNA sequencing data. And some of these efforts and some of these models are at least partially applicable to what we are doing, and therefore we have to learn, but not just take them as wholesale at face value, but adapt them in an intelligent way. And that's why I have invited to this meeting some of the colleagues who have designed such methods for single-cell RNA sequencing data, so that we can begin the conversation of how do we build upon these existing efforts to come up with something that really is well suited for the kind of data that we generate. We talked a lot about missing values and imputation and whether we should do it and whether we shouldn't do it. And I want to go back to a point that Nathan made earlier today, that the distribution of abundances of proteins that we did not quantify is different from the distribution of abundances of proteins that we did quantify. And that is important to take into account. Usually imputation methods don't do that, and that's why we are developing such a method in collaboration with Alexander Franks, where we want to take into account these different underlying distributions. Another thing is we have estimates for population average, bulk levels of these proteins that have missing data. These data points are actually very informative about the nature of the missing data points that we did not see. We want to incorporate this. Another unique feature to our data when it comes to imputing missing values is that there would be certain cutoff on the signal-to-noise ratio of the signal that mass-spec instruments record and that we use to quantify peptides. But there might be values below that cutoff that are still useful priors if we're going to impute the data. So all of these things are worthwhile taking into account, thinking very hard to begin with, what is the generative process? Why are we missing the data to begin with, so that these kind of considerations and thinking can inform what is the best way to impute the missing values. And there is a lot of analysis that we can do without doing the imputation to begin with. Even without doing imputation, analysis may still assume certain things for the values that were not imputed. Now, overall, in our data, in the data that Harrison presented, and I'll talk about more tomorrow, we had about 2,000 proteins analyzed across all of the cells, and we had about 1,000 quantified per single cell. So we had about 50% missing data, which is very small compared to typical single-cell RNA data set, where it's much closer to being 90% missing data. So it is many, many folds lower level of missing data. And to be able to take full advantage of these opportunities to develop good computational platforms, we need to engage a broader community, people who would not necessarily want to analyze their own mass-spec data. And therefore, I find it very useful to provide data in formats that are easy to be used by computational folks in the community, those who don't want to run the max-quant searches. So it goes without saying that overall data should be deposited in the conventional community databases such as Massif and Proteome Exchange. But in addition to that, I think it is very useful to come up with formats that make it easier for people, both computational groups and biology-oriented groups, to be able to more easily reuse the data. And this meeting is an opportunity to discuss what these formats are. Now, some formats that we have decided to share the data are just matrices of peptides versus single cells. data that are as close to the search engine results as possible without any batch effect corrections, no imputation, nothing, just a tab delimited file that can be easily transported and shared with computational groups that may want to work further on this data. Another one would be the protein levels processed, perhaps batch corrected, imputed to a certain degree, which would be very easily analyzed by biology groups who just want to see what is the result based on this analysis. And of course, these data cannot be meaningfully analyzed without all of the associated metadata about the single cells. All of the information that we have, it could be the abundance of fluorescent proteins that we measured, it could be their size, it could be their granularity, it could be their isolation, a myriad of other useful features that we record and that are provided alongside. So you can already download this format of data from the Scope2 website. Harrison has very nicely prepared these files. This is the HTTP URL from which you can download the data. And I'm very open to your feedback and ideas as to what are other data types that you might find useful, or what are some modifications to these data types that you would like us to introduce. So moving to our metrics and quality standards for evaluating the data, there are at least two conceptually distinct levels of doing this evaluation. One is if Joe and Elizabeth have the same sample, are they going to produce the same single cell data? That's a really important benchmark to have, and one that currently is hard to have because few laboratories are analyzing the same samples. And I want to start a conversation as to how we get there. Ideally, a company can, one of the vendors can distribute such samples. In the short term, I'm certainly very happy to send any one of you some of our 1x master samples that Harrison, Grant, Ed described in details, so that you can analyze on your LC-MS system the samples that we analyze and directly compare your results and see, is your system single cell ready? Do you get better results? Do you get worse results? And I'm not going to talk much more about this, even though it's a very important thing to get going, and it's going to be one of the things that you'll discuss more at the end of Wednesday. What I'm going to focus much more on is what are some quantitative metrics that we can use to evaluate data and convince ourselves that what we quantify in single cells is useful for biological analysis. Do we see patterns that are fiction of our imagination, or do we see patterns that reflect genuine underlying biology? And it's much easier to start with saying what doesn't work than saying what does work. So I'm going to start with what I think doesn't work. Here is one example of that. We can compare the absolute intensities, abundances of two proteomes, and compute a correlation. In this case happens to be 0.95. So what kind of inference can we make based on this? It's high correlation, so it seems like these two proteomes are similar. Does it mean that our measurements are accurate? So let me help you and tell you that these two proteomes come from two different cell types. They're actually quite different, both immune cell types, but they're different cell types. And you may say, oh, well, maybe T cells at the proteome levels are similar to monocytes. That's why we see this high correlation. And that's not true. Actually, there are proteins that differ in their relative abundances between T cells and monocytes by an order of magnitude. But this order of magnitude is relatively small when plotted on a 5 order of magnitude scale. And this is not true just for monocytes and T cells. This is in fact true for almost any human tissue that we may want to analyze. Here are scatter plots of protein abundances measured as part of the draft map of the human proteome, from kidneys, from stomach, from all sorts, from brain, from muscle, from different tissues. And if you correlate just about the proteome of just about any tissue to the proteome of just any other tissue, you'll find them to be highly correlated. Why? I can tell you. So these proteins here at the top, they're the histones. Right below them are ribosomal proteins. Right below them are the mitochondrial proteins and the cytoskeletal. So this kind of comparison in the global scale is a very dull measure for anything. And the only conclusion that I'm willing to make based on comparing these two samples is that they're human samples. We cannot really conclude that we have accurate quantitation. Now, there's another reason why we cannot conclude that our quantitation here is accurate. What is that reason? Let me help you. Replicability is not the same as accuracy. So if you just compare the measurements obtained, even from different biological replicates, you can get those to be very similar, which doesn't mean that they're accurate. Here is a plot from an early Nature paper, very influential Nature paper that measured protein levels by GFP-tagged proteins. And they reported this astoundingly high R-squared, which creates the impression that you have a very accurate measurement. But again, all that this shows is that the measurements are reproduced. It doesn't tell you that what you measure is correct. And in fact, in this case, we can fast forward to 2018 and look at this meta-analysis of protein abundances measured in budding yeast using different methods, including lots of groups using flow cytometries and groups using mass spectrometry and groups using different methods. And if you compare the scatter plots here in this part of the plot, they look less tight than this one. So this one showed you what is the random measurement error. It did not tell you anything about systematics. There are a lot of systematic errors that can be highly reproducible from sample to sample. And therefore, this kind of plot is no good for being able to measure accuracy. It is good for being able to measure reproducibility, but not accuracy. So what can we do for that? I don't have a panacea. You're going to have systematics in your data. We have systematic errors in our data. But the one thing that I have found incredibly useful in our work, and one thing that I always aspire to, is to incorporate as many controls in the experiments as possible. And how does this work? It works by having samples that are processed simultaneously, that see the same surfaces. You can think of a Silac workflow where you can do the digestion together, or you can think of a TMT workflow where you can do the labeling, and at least after you mix the samples, all of the systematics due to different LC conditions, different ionization, different fragmentation, all of these things can be controlled for. So there's no panacea. Nothing's going to eradicate once and forever systematic errors. And therefore, we have to be cognizant that they're there and not mistake reproducibility for accuracy. And I think it's also good to aspire to try to minimize them. And I think we have great ways of minimizing them by doing controlled experiments, by using ratiometric measurements rather than rely on absolute intensities that certainly here are not well reproduced. All right, so can we use dimensionality reduction and separation in t-SNE plots or any other kind of plots to validate the accuracy of our quantitation? You can kind of guess the answer here, but can you give me a reason? Why don't I like them? Now, in the case of t-SNE, anybody who has used it, or UMAP, would know that the result depends on some parameters that are being set. And by changing the parameter, you can get different clustering. And in fact, on a t-SNE plot, if you don't know, you should know that the distance between the blue cluster and the orange cluster being large has nothing to do with these two clusters being more different than the orange to the pink. The large distances are simply not informative. But in addition to parameters that can result in clustering based on differences that are not quite so clear. Let's say you don't know what fraction of the variance is actually used to delineate these clusters. But let's say you used PCA. Let's say that there are no fudge parameters, and you found that your principal components explain a large fraction of the variance that you're plotting. Can you use a separation to declare a victory? No. You certainly cannot, because it could be that you simply failed to lyse one cell type. That cell type was particularly hard to lyse. Easy to separate. You don't separate based on useful biology. It could be that you have batch effects. Batch effects can really well separate cell populations. Well, they do separate them, but is that useful separation? So let me transition to some measures that I think are more useful. And again, I want to have this as an ongoing discussion and a dialogue rather than a dogmatic statement that this or else. But some of the measures that we have found useful is comparing diluted single-cell lysates, relative quantitation derived from diluted single-cell lysates, to relative quantitation derived from conventional bulk proteomic methods. It could be Silac, it could be TMT, it could be another measurement. Now, why do I like the diluted cell lysates here? There's no biological variability. I know that in the absence of any noise, I should get identity. I should get exactly the same ratios. To the extent that I don't, that reflects measurement error. What about the systematics? Well, by taking the ratio of samples that have experienced the same workflow, especially if these are Silac samples that have been digested together and analyzed together, we can control for a lot of the systematics. And generally, I have found these measurements to be very useful. And they also support the possibility that LC-MS instruments are able to do accurate quantification at protein levels present in single-cell lysates. And let me compare this to single-cell RNA-seq. So here on the right, I have an example from one of the best methods for single-cell RNA sequencing, SMART-seq2, from a relatively recent Nature Biotechnology paper. It's still one of the best methods, actually. It's more accurate than the high-throughput droplet-based methods because it allows for higher sampling. And the quantitation of these relative ratios appears to be less accurate than what we can do with proteomics. And here you have the dilemma. If you look at the single-cell protein measurements, they're not as accurate as measurements from bulk samples. We don't have as high signal-to-noise ratio. We don't sample as many ions. But they're certainly more accurate than what we're able to do with just counting a few transcripts in single cells. And I also think they can be made much more accurate, both by advances in instrumentation, changes in the methods. These are just our early baby steps that are already giving quantitation, relative quantitation, that is superior to that of mature single-cell RNA sequencing methods that are a billion-dollar market. And I find this very exciting, very much along the lines of what Harris was saying. So if you wanted to validate the ability to identify different cell subpopulations, one thing that I have found useful in our work is, in addition to the markers corresponding to single cells, to plot samples that correspond to well-validated measurements, such as bulk samples. Now, in this case, on this PCA plot, we plot not only data points from single-cell lysates diluted to single-cell level, but we also plot with pluses the corresponding samples from carrier samples or from Silac experiments, for that matter. And what gives me confidence that this separation in PCA space is not due to differential cell lysis is that the bulk samples separate the same way as the single-cell samples. And then I'm going to return to this theme of using features that are specific to bottom-up proteomics, of having multiple measurements for the same protein deriving from different peptides. We're all familiar, at least all of us who have played with mass-spec data, we are all familiar with these measurements, and they can be a reasonably reliable and easy metric that can be applied to any data set without doing anything further to evaluate what is the reliability of those measurements. And in this case, this is a figure taken from a recent paper that Albert Chen, a student in my group, developed and published on being able to increase the confidence of identifying peptide sequences with insufficient MS2 fragments to support confident sequence identification. And in this case, Albert went into extensive validation to establish that these peptides provide accurate quantification. Peptides that are identified based on DART-AD have CVs, coefficients of CVs, that are comparable or lower than these identified based on spectra alone. But he's going to tell you much more about this work in more details on Wednesday. Then I'm going to return to this measurement based on external methods, because it's the ultimate cure for systematic errors. One very good way to guard against systematic errors is to have a completely different, completely orthogonal measurement. And this kind of measurements are very hard to do in single cells. They're very hard. But I think it's still worthwhile using them. And in this case, we have found fluorescent proteins to be more reliable measure for quantitative abundances of proteins and antibodies, because they don't have the problem of specificity. And I think that's a measure that we should continue to use. It certainly has been useful for us. These are experiments that Guillaume Harmonich, a student in my group, did. And as I said, I share our thoughts. I did not tell you all of the useful benchmark metrics that we can be using. These are some of the metrics that we have found useful and that seem good to me. But I would love to hear what are your ideas, what are additional metrics, or what are additions to these ones that you would like to make. And we are going to have a discussion session devoted to discussing these metrics and other questions on Wednesday. So I would like to encourage all of you to think about these questions and bring your suggestions. Bring your suggestions for questions to discuss on Wednesday, and bring your suggestions for what might be good metrics to be used for benchmarking quantitation in single-cell proteomics measurements. I think now is the right time to establish those metrics, because while the community is still small and we can have most of the people engage in doing these kind of measurements in one room, we can agree on those, maybe write some white papers that then can become widely adopted and used by the community. And we prevent some very embarrassing mistakes, as happened in single-cell RNA sequencing. Many of you might be familiar that the first few dozen high-profile single-cell RNA sequencing paper mostly separated their cells based on missing data, rather than based on any relevant quantitation. And we are prone to do the same unless we have reliable benchmarks that can help us know which patterns are false and which patterns are reliable, what comes from the biology, what comes from the data. So I would very much like to hear your input, your thoughts on what are good metrics that can guide us and bring us closer to biology and further away from systematics. And I want to finish by thanking everybody in my group and beyond who has contributed to even to the little data that I showed today, and for making this possible, not just in terms of, I should say, not just in terms of scientific contributions to our work, but also to help organize this conference. This has been a wonderful group effort from my group and beyond, many students from Alexander Ivanov's group, Bogdan Budnik from Olga's group. Thank you so much for helping organize the conference and for making this work possible. So I'll take any questions and any suggestions.
So thank you. So you use the Silac as one of the benchmark methods, right? So you use Silac.
Silac,
yeah,
yes, we like Silac. I love Silac, actually.
But why Silac?
Why Silac?
Yeah.
Because it allows me to control for digestion variability.
Okay.
And because it allows me to do quantitation that is based on different ions compared to TMT. And every time I can change my experiments to have a different measurement, I like to take that opportunity.
Now that you have isobaric MBR match between runs, I think maybe it's more inconvenient to use TMT itself.
Maybe it's more inconvenient to use TMT?
More convenient.
More convenient to use
Instead
TMT.
Of the Silac is because it's completely a different system. And actually, the first time I read your paper about Silac, I thought you were going to use Silac as one of the branches of scope mass.
Well, the important thing is that we believe there is a true ratio between the relative protein abundance in, let's say, two samples. And that relative protein abundance should be the same whether measured by Silac or measured by TMT. If it's not the same, then we have a problem.
Okay.
And Silac is very useful for me as being a measurement that is fairly different from the TMT measurement while still being high throughput and accurate, that gives me a clear benchmark that helps me stay on the path of data rather than on the path of hallucination.
Okay, but you know what? Actually, with Silac, you can minimize the sample loss. You know, at the very beginning instead of at the peptide level. So maybe it's also an alternative route. I'm not sure.
Yeah, Silac certainly has its own advantages. Can be used in a variety of different ways.
Thank you. Great talk. I want to add a note that for the standard quality control sample, Thermo Fisher has commercialized a standard called the East 11-plex sample. Maybe we could distribute, maybe we can buy from Thermo Fisher, though it is a little bit expensive, and we can add some controls, maybe HeLa. So we all start from the same commercialized sample. It's easy to distribute in different countries, I guess.
Thank you. Thank you. That's a good suggestion.
Might be less complex than human sample. This might be a problem.
Well, I think that we'll have to design a sample that contains at least some of the features that we find important, such as the ability to reliably benchmark or relative quantitation. For me, that's very important. So we'll have to work with them, come up with a sample that ideally then can help us dispatch to different labs. But thank you. I think that's a good suggestion, and we should continue discussing it.
From my experience in bulk amount samples from the East 11-plex, if you do TMT MS2 measurements, it's very difficult to get a precise quantification. From my experience, maybe when the chromatography gets better, the ratio will be much better. Yep. You can look into their manuals and find the results. There are some results published by Gigi's group in Harvard Medical School.
Yeah. And that's why it will be wonderful to have a consistent benchmarks used by everybody so that we can compare and see how good quantitation really is. There is also the point that I should say here that one might be able to make important and useful biological inferences, even with imperfect quantitation. I think that the aspiration to have precise quantitation and very accurate quantitation is wonderful, and we should pursue this. But there is the aspect that if your data are 80% reliable, doesn't mean they're not usable. You can still use them. You just have to be aware and computationally savvy to use them in the right way so that you end up making correct inferences.
Thank you. I have another suggestion for, it's not necessarily quality control, but it's like for a carrier channel or carrier proteome, because I mean, most of us are working with human cells, right? And also utilizing the carrier proteome within an experimental design. So if we combine a number of available human cell lines and generate a more or less universal carrier proteome internal standard or something that we can benchmark against a various number of human cells or cell types. That would be, I think, perhaps a good idea to start with. And that can also allow us to normalize more or less number of proteins, at least within this human cell lines, maybe boosting some low abundant or lower abundant proteins in a given cell line.
Thank you. I think that's certainly feasible. Going back to this plot here, my computer is willing to cooperate. So going back to this plot here, because the overall abundances of proteins is not so different, it actually means that even if you're analyzing T cells, you can make the carrier from monocytes and still get data. In fact, this is precisely the experiment that we did as part of a request for reviewers for our SCOPE-MS paper. In that case, we used the HEK cells as carrier to be able to distinguish T cells from monocytes, and that worked well. So I think it's feasible. One can certainly make a representative carrier. I'm not, maybe more for benchmarking purposes rather than for each experiment. I think that if I'm doing a biological experiment, I would still try to choose a reference that is more appropriate for the single cells that are part of my biological experiment. But I think the idea of having a standard carrier as part of our benchmark sample is a very good one. Thank you.
Can you elaborate a little bit on the last sentence, the comment you made about the RNA-seq, that they were separating cells based on the missing data? How did the field realize they were making that mistake, and how did they fix it?
Oh, that's easy. So with what happened there is that the efficiency of analyzing each individual cell by RNA sequencing is different. From some cells, you're going to get more RNA. From other cells, you're going to get less RNA. When you get less RNA, there will be more missing values. And that's the state of the data. There is a distribution as to how many missing values you have per single point, per single cell. That is also the case actually with SCOPE-MS data, but to a much, much, much smaller degree than with single-cell RNA sequencing data, in part also because we remove cells that do not give us very good data. But that certainly was the case in the early experiments, continue to be the case now. And if you do the principal component analysis, which they did, and look at what features of the data the principal components correlate to, then you find that the correlation between the fraction of missing data and principal component 1 is 0.98. Essentially, your first principal component is perfectly correlated, perfect indication for how much missing data you had. Now, if you use your first principal component to separate cells into distinct clusters, you'll be separating them based on how much missing data you had in each cluster. And then you'll end up with two distinct clusters that can make a nice plot for a Nature paper. But that separation is driven by success rate of your sample preparation rather than by any interesting biology worthwhile the attention of the community. And that was made clear by reanalysis of the data by multiple groups, and now has been well established. I don't think it's widely discussed, even though the evidence for that is very clear and it's well acknowledged.
Just one comment. Also, in the recent papers, they're talking about now normalization based on the cell cycle. Because again, in the earlier papers, they realized that they're actually clustering based on different stages in the cell cycle.
That's right. So in single cell measurements, normalization turns out to be a very tricky business. And there have been a number of high-profile papers on that topic for single cell RNA sequencing. And I don't know that anybody has found the perfect solution for that. It remains a difficult issue. One thing that I actually quite liked, even though I don't have much experience with it to say how successful it will be, we played a little bit with it, we used it with our data, was this idea of using, what was it called, the ratio measure as a pairwise similarity metric. Instead of using Euclidean distance, Pearson correlation, mutual information, and so on, the more familiar metrics, you can just use the log of the variance, of the covariance of two mRNAs or two proteins, and that has some very good properties that are independent from the normalization that you use. That was a recent Nature Methods paper that I'm not describing in great detail, particularly well now, but I'll be happy to give you the reference offline and talk more about it. One more question from Sophia.
Yes. You talk about DDA data, but what about DIA data?
Oh, you're going to talk about it tomorrow, I hope. I think it's great. I think it's a different approach from what we are pursuing. I would actually love to do data-independent acquisition once we do our quantitation from the complement ions rather than from the reporter ions. I would love to get there. We're not there yet. So with TMT, currently there is no feasible way to make the data-independent acquisition. And TMT provides some substantial advantages for being able to multiplex the measurements. For me, one of the key points, which is so obvious that we rarely talk about it, we didn't talk about it much today, is the multiplexing, because mass-spec instruments are marvels of engineering. They're expensive, and they will not get cheap. The only way to get single-cell proteomics to be very useful for biology and to produce a lot of data at a reasonable cost is to have multiplexing. I'm not happy with the 11-plex. I think it should be 100-plex. I'm trying to convince colleagues to work on that. It's entirely feasible. I actually have some interest in doing it, but not too actively. Haven't gotten there yet. But I think we should do that. I think it will be great to do a isobaric reagent that allows us to do data-independent acquisition, where it produces complement ions more efficiently. And I know there have been efforts in that direction. I think they don't quite solve the problem yet. But that allows to do data-independent acquisition with isobaric labeling. And then we are going to have a bonanza of awesome data. At the moment, there are certainly some clever approaches that might take advantage of data-independent acquisition. And I'm very much looking forward to learn more about what you're doing in Rudy's group on that front. All right. Thank you very much for attending the workshop.